Okay friends, now we are going to talk about a very complicated and as well as interesting topic, protein folding and protein stability. Now before going to the discussion about protein folding, I have previously discussed about protein folding in my previous videos, which is a basic understanding, but in this lecture I am going to focus on some very important points about protein folding. Before knowing the protein folding thing, we need to know about the protein stability and how a protein structure is made. So first things first, we have to talk about the primary structure of a protein, because as we know, what is a protein? What is a protein? Protein is nothing but a polymer of amino acids. So it's a poly amino acid sequences. That means a protein. Now amino acid, suppose this is amino acid A, this is amino acid B, this is amino acid 2, amino acid 3, and all these amino acids are joined with each other via the peptide bond, and they will form linear stretches of amino acid sequences like that. If I draw this sequence like that, and this is the backbone one, this is the N terminal of this backbone, this is the C terminal of the backbone, then we are having the linear structure of a protein, and in this protein we are having this amino acid sequences. So this dot, this lines, small lines represent that amino acid sequences. Now few of those amino acid sequences are hydrophobic in nature. So they are hydrophobic in nature. Hydrophobic means they just hate water. They want to go against water all the time. They do not want to shake their hand with water, and they are a hydrophilic amino acid sequences. And those sequences actually love water, so they just love to interact with water. So this hydrophilic sequence and hydrophobic sequence, both of the sequence present in this primary structure of this protein backbone. Now if we think about the stability of a protein, now what are the forces that makes a protein really stable and rigid in nature? Because protein is the most versatile thing in inside a cell, and protein is the most important part inside a cell, as we know, because protein have to do all the works and all the work to be done by the proteins inside the cell until we live on. Okay, now that's why you need to have these proteins really structurally rigid, structurally favorable in nature. And which things actually denote these things, this feature of the protein? Now there are several bonds. The answer lies in chemical bonding. So what type of bonds we can find inside a protein? Now the first or primary bond that we can see inside a protein, this is the protein backbone bond, that is called the peptide bond, which is the main force or main organic bond that that hold these amino acid structures together. Okay, and then there are other forces like the hydrogen bonding. So there are hydrogen bonds, there are electrostatic bonds, there are hydrophobic interactions, there are salt bridge interactions, and all these interactions are really important for holding the structure of a protein together. Now and how this primary sequence of the structure finally make a protein structure? Because in normal situations we can see the proteins most of the time are globular in nature, sometimes they are fibrillar in nature. Okay, now how these primary stretches of amino acid sequence really end up with a structure like this globule or this fibrillar structure? The answer lies on the interaction between amino acid sequences. As we know, there are hydrophilic amino acid. Those hydrophilic amino acid can interact with each other. They can interact with water also. And there are hydrophobic amino acid. They can only interact with other hydrophobic amino acid. They cannot interact with any water or water solvent. Okay, so the hydrophilic is going to find hydrophilic, the hydrophobic is going to find hydrophobic, and there are charged residue of amino acid sequences. Those charged amino acid sequences, they are also part of the hydrophilic residues. Now these charged residues will interact with each other. The opposite charge will interact with each other. So a positive charged amino acid sequence eventually going to interact with the negative charged amino acid, and they can finally make some structure like this globule or this spiral. Okay, so this amino acid sequence or stretch of amino acid sequence, which is the primary structure of the protein, there are amino acid sequences which are interacting with each other and finally make a structure like that. Now these are the stability. So chemical bonds give this protein not only chemical stability but also the physical stability. So whenever we are talking about the protein stability, we are actually talking about chemical as well as physical stability, and most importantly we are talking about the physical stability, because that is the important thing for us for a protein to function properly. that has to be physically stable rather than the chemical stability. Okay, now as we see in this picture, which is the summer up view of the protein folding, that we are having this primary stage of amino acid. Now the amino acid sequence start to interact with each other, and when they interact, they are interacting, and finally they lead to formation of some structure. After forming some particular structure, they further they are not modified, so they are just stay as it is for some times. So whenever we just start to form a protein structure like this primary sequence of amino acid stretch, that is not stable, that is not favorable, so they start to interact with each other, the amino acids start to interact with each other until and unless they reaches their favorable structure, which is in this picture is this structure. Okay, so that is called the protein folding. So we are having the amino acid sequences together linked by peptide bonds. We are having the primary sequence of amino acid. Now this primary sequence of amino acids are interacting with each other, they find different ways to bond with each other, and finally until they reach their desired structural configuration, which is in this case we can see in like that. Okay, whenever it reaches this configuration, they just stop doing anything, they just start to function on their own now. Okay, and the structure is really important. Why? Because the structure defines the function. As I've told always, the structure defines the function, because if we modify the structure from one to another, then the function of the protein will definitely going to change. That's why the structure is really important. Sometimes we need the protein to be granular, to be huge, and sometimes we need the protein to be spiralum to make a channel, okay, inside the cell membrane. So depending upon the presence, depending upon our need, the protein structure will vary. Now if we talk about the thermodynamics of the protein stability, because thermodynamics is the term, the subject that helps us to define whether something is stable or not using the free energy. Now as we know, what is free energy? Free energy is the energy for doing a particular job. Now as we know, according to the Gibbs equation, that this del G or free energy change for unfolded and folded state, if this change is negative, sorry, if this del G, my mouse is again troubling me, this is very bad, you know, I have very hard time to write anything inside this. So we have a del G here. Now if the del G is negative, then that means this process is spontaneous and can easily be occurred. If del G is positive, then this reaction is not favorable. Now in this protein folding thing, if we look for that, if a protein have to be folded properly, what is the driving force for that? And as we know, any situation inside this environment, if we think about the thermodynamics, three things really matter. One is this entropy, another is enthalpy, which is the internal energy change, another one is this free energy. Now in this case we are going to focus on entropy and del G. Now as we know, according to this del G equation, the equation says us something like that: del G equals del H, this, that means the enthalpy change, and minus T del S. So this is the equation of Gibbs. Now by looking at this equation, we can say if we are increasing this S, that means the entropy, we are actually making the del G value negative, and that means the favorable reaction. So if in the environment entropy is increasing for any reaction, that reaction is going to be favorable in nature. Okay, so we have to have del S negative, we have to have del S positive. So we need to rise this del S, and rising this del S will help us to make this reaction favorable in the environment. So and what is entropy, by the way? The entropy is the measurement of what you can say disorder in the nature. So there are disordered conditions and ordered conditions, messy conditions as well as the ordered condition. In the messy condition, the entropy is higher. In the ordered condition, the entropy is lower. Now if we think about amino acids, so we have amino acid sequence, lots of amino acid sequences there, and we are having amino acids lie together to make a primary structure of the protein. Then we are having simple complex structure of the protein. Then we are finally having our 3D structure of the protein. Now in this different arrangement of protein folding, which one is having the highest entropy? The answer is definitely this one, this amino acid sequence, that is the highest entropy gaining region or gaining state. and this folded state is having the lowest entropy change. Okay, that means if we are talking about the only amino acid sequences in the environment, they are having the higher entropy, and what we are trying to do in this protein folding, we are actually incorporating this amino acid to make a stabilized structure. We are actually making this system more stable from the random orientation. So we are actually decreasing the randomness of the environment. That means we are decreasing this entropy, and decreasing entropy will not be favored in the environment according to the law of thermodynamics, according to the second law of thermodynamics. So why and how the proteins start to fold, and what actually helps them to fold properly, and what actually holds this folded structure of the protein against the law of thermodynamics, that is the biggest question. And the answer lies between the nature of protein. It lies between the interaction that protein subunits, that amino acid sequences will take by doing their own interactions with themselves. We'll see in future slides that. Okay, now if we look about this thermodynamics, then you can find this is the del G, or the free free energy change, and this free energy change for the folding of a protein will be the free energy for a native state minus the free energy for an unfolded state. Now if this is negative, that means the reaction will be spontaneous, and the way of making this spontaneous, the way of making this equation negative, is try to increase the free energy for unfolded state and decrease the free energy for the native state. So if we can do this, that means we can make this protein to be folded properly according to the thermodynamics law. Okay, so how we are doing this, and why not why actually, how we are doing this, how the cell make the way to establish these things inside it. Okay, let's find it out. Now if we think about the importance of the protein stability, that means the increased protein stability may be multi-billion-dollar value in the food and drug processing. That's why we are actually think about this protein stability most of the time, because if we can make if we can understand about the protein stability, that we can stabilize a protein for much longer time. We can store a protein for much longer time. That will give us save us $1 million money actually. That's why we are very much interesting about the protein folding, as well as scientifically, as well as different economic values, economic perspectives, because in drug industry we need that thing. Now if we think about the stability of the folded state, then what we can find here, we have in the folded state stability, we have to measure the protein stability between difference between the unfolded state and the folded state. So we need to measure the free energy change or free energy difference. So how can we measure? Del G equals del N native minus del E unfolded. We can look at the structure, and we can look here, it is something this. So that gives us a very shocking actually very shocking thing, that is in aqueous solution at room temperature. If you use aqueous solution and then room temperature, the ratio of the folded over unfolded protein is 2 into 10⁻⁷:1. That means inside the room temperature and the aqueous solution environment, the unfolded state or chance of finding an unfolded protein is extremely low. You can see the chance of finding the folded protein is 2 into 10⁻⁷, but chance of finding an unfolded protein in the same situation is only 1. So we are having 2 into 10⁻⁷ folded protein and only 1 unfolded protein in that aqueous system. So that means proteins always wanted to be in the folded state. So just these are the actual take-home messages. So just remember this take-home messages, what I used to tell you, and what I am going to tell you in the future slide. So proteins always tend to be in the folded state. They want to be in the folded state rather the unfolded state in the aqueous environment at the room temperature. If we increase the temperature, these things may not be possible. If we just change this solution from aqueous to another type of solvent, then this thing will be hampered. But if we are having the aqueous solution, if we are having in the room temperature, the protein always wanted to be in the folded state rather the unfolded state, because of their own interaction, because of the amino acid interaction, because of the different types of hydrophilic amino acid they possess, because of the different hydrophobic amino acid and hydrophobic interactions they can do. Okay, now let us talk about this stability of folded state again, and if we think about here, that a very interesting thing is given. Now K is an equilibrium constant for this folding and unfolding. So suppose we have the protein here, protein is folded, and we have the protein unfolded. So this is our reaction. If we just think about the reaction of protein folding and unfolding, then we are just going towards the unfolded state of the protein from folded state of the protein, and the rate constant for this reaction is K, capital K, and capital K equals to here Kf divided by K sub U. Now this is the rate constant for this protein unfolding and folding, and what we can find here, if a typical protein refolds spontaneously with a rate constant of Kf equals to 1 per second, then the rates of spontaneity unfolding under the same condition will be 10 to the power minus 7 per second. That means the half-life of this protein unfolding state is 80 days. So just try to think about this again. Now, if a typical protein refolds, that means we are having a protein. Suppose we are having a protein. Now we unfold that protein. Folded state, this is the folded, so we unfold that protein. Now again we try to refold that protein and make it to reach the folded conformation, and then what happens, that the spontaneity for making the refolding of the protein is almost 1 per second, but its rate of spontaneously unfolding from the folded state is 10 to the power minus 7 per second. So that means the half-life is almost 80 days. So protein can fold really quick, as we can see in this case. Okay, so the unfolding need 80 days for this purpose. Okay, for the at the same condition. So that is a very important assumption in this case. Okay, so that again suggests us the unfolding of the protein will only be a transient state. So unfolding is a transient state, but the most of the time the protein is to be presented in the folded state. That is the desired state of their nature. That is their nature to be presented. Sometimes they need to go through that unfolded state. So unfolding or unfolded state of a protein itself is not at all a state. This is a transient state. This is not a stabilized. So we can say this again. And only this unfolding state or unfolded state of the protein is just a transient state between the folded state. So we have a folded state, we have unfolded, then refolding. So this is just a transition. The unfolded state is just a transition about that. Now what are the different things that can unfold a protein, that can make a protein unstable? First thing is pH. Extreme pH can make a protein unfolded, because that the net charge of the protein due to the titration of all the ionizing group leads to the intramolecular charge repulsion. If we change the pH, suppose if we increase the pH, we start to increase the pH, that means we are actually making the hydrogen ion concentration getting lower and OH minus concentration getting higher. So it actually going to affect the amino acid, the charged amino acid sequences, because we are having the charged amino acid sequences in the backbone. They will eventually going to titrate the amino acid charged amino acid sequences in the backbone. They are going to ionize the group, those amino acid groups, and that leads to the intramolecular charge repulsion. So if we increases the pH, it will incorporate that charge changes inside the protein backbone, that will eventually going to repel each other, and finally the backbone will be destabilized and the protein structure will be disrupted. So that is the effect of pH. Now if we think about other denaturants, like here we have urea, we can also have guanidinium ion, we can also have mercaptoethanol and all these things. Now we can use these things to actually denature a protein backbone. Now both side chains and backbone appear to be more stable in the presence of the denaturant. Okay, so if we add this, there is no very good solvent. That is another very good assumption. If you think here, there is no very good solvent there which can stabilize a protein structure. Why? Because solvent are a particular one type. So either the solvent we are using is hydrophobic, or it can be hydrophobic, which is our organic solvent. And because of that, protein structure is having both type of sequences, hydrophobic and hydrophilic. Now if we are adding the hydrophilic proteins in the hydrophilic solvent, that can be stable. If we add a protein structure, globular protein, into a hydrophilic solvent, so we have a hydrophilic solvent. So we have hydrophilic solvent, and this hydrophilic solvent we are adding a protein. Now protein is possessing both hydrophobic and hydrophilic both. Now this protein is going to interact with this hydrophilic solvent. Now the hydrophilic sequence of the protein will interact with the hydrophilic solvent, and they can stay as it is, they can stay very comfortably. But the hydrophobic sequences of this protein will try to go against this solvent, and that's why the protein need to be folded properly in these situations. So that in this case the hydrophobic will make trouble. Now if we add this protein in the hydrophobic solvent, then the hydrophobic sequences stay comfortably, but the hydrophilic sequence will go against this hydrophobic environment, and they are going to trying to put themselves in the interior side of the protein. That's why the protein need to be folded. So that's why in the solvent situation, so whenever we are providing any solvent, we can say that means we are actually giving them a force, giving them an indirect force to make this protein stabilize via making the protein folding, because they need to fold. Otherwise the protein residues will be exposed, the amino acid residues will be exposed to the environment. That is not at all that will not be at all favorable at the environment. Okay, now if we think about the protein stability depending on the presence of different ions, as you can see here, so SCN, these are the negative ions. So you can see the sulfate ion will give the highest stability of the protein, because protein backbone will have the different charges, and the sulfate will interact with that positive charge. There is urea, then chlorine, there is SCN. So these are the different ions which actually helps the protein to stabilize in environmental situations. Okay, now you can disrupt the protein structure by using these ions also. Okay, because if we use them against the same type of amino acid sequences, these are all negatively charged ions. Now if we use these negatively charged ions more and more negatively charged ions in the amino acid sequences where they are having the negative charged amino acid sequence, they will disrupt those sequences in the protein backbone, and the protein backbone will be cleaved off. Now if we think about the thermal denaturation, and the denaturation or the protein instability due to the heat, that is a very important, because heat can do anything, as we know. Heat can disrupt all the different types of bonding, including the hydrogen bonding, and also it can increase the hydrophobicity. As it disrupts the hydrogen bonding, it can expose more and more amino acid sequences outside the environment to face the solvent, and that is again increase the hydrophobicity of those hydrophobic amino acid sequences, and they try to come together, come nearer to arrange themselves in the hydrophobic collapse, and finally make another type of protein structure to go against the del G change. Okay, now there are some thermophilic proteins that we can find in some bacterial cell, which can live in a hydrothermal vent, which can stay at over the 100 degrees Celsius temperature. How this protein manages to survive over this 100 degrees Celsius temperature? The answer lies not only in their three-dimensional structures, because if we think about the three-dimensional structure of normal protein as well as the thermophilic proteins, we can hardly find any difference. There is no difference at all in their three-dimensional structure. We can find essentially this stabilized or same type of structure in case of thermophilic as well as the mesophilic proteins, but the difference is on the hydrogen bonding. So they just change their hydrogen bonding. There are more hydrogen bond present in the thermophilic protein rather than the mesophilic protein. So they need much heat to destabilize this protein structure, because there are much more hydrogen bonding. So they need much more energy to break this hydrogen bond to make the protein unstable. Okay, and also there are the presence of different type of amino acid is also important, because in thermophilic proteins we have seen the increased amount of arginine and an occurrence of alanine in their helices. Okay, so normally if we think about the mesophilic protein, in the mesophilic protein helices we can find many alanine residue, but in case of this same type of thermophilic protein, so we take one mesophilic protein, one thermophilic protein, and this both of the protein are supposed in helix. Now this mesophilic protein helix contains the alanine, and we have the same three-dimensional structure of a thermophilic protein, which is also an helix, but those helix is consisting of more arginine residues instead of the alanine residues. Okay, so this residue variation is also giving them the stability at these higher temperatures. Okay, and also we can find the increased number of salt bridge interactions, because they are possessing this thermophilic proteins are possessing more and more charged amino acid sequences, like lysine, like glutamic acid, aspartic acid, like histidine, arginine, and all these amino acid. And these amino acid sequences will interact with each other via salt bridge interactions. The salt bridge interaction, like from with lysine and glutamic acid, can hold this structure really to hold this structure together for a long time. Okay, and sometimes they also provide some chelating agents, some metal ions, which can stabilize the structure, because if we are having the negative structure of the protein backbone, then if they incorporate the calcium or the bivalent cations, then they can stabilize this protein structure. Now another very important assumption here is the stability and activity thing. Now suppose there are protein sequences, we have the primary sequences of amino acid, we can fold them to make a protein structure. This is the protein structure. We can also fold them to make a protein structure, which is this. So these are two different types of 3D protein structure we can form from this one primary amino acid sequence, amino acid arranged or polyamino acid sequence. But if we look at this one, this is the first type of structure, this is the second type of structure. And if we go on and produce this first type of structure, this is very active enzyme, and this enzyme functions very properly and very actively. But in the second, if we form a structure like this second type, it will not much active. This is not at all a very active enzyme. The activity of the enzyme actually falls. But the question, but another important thing is but this protein, this second structure, is much more stable. So this is much more stable than this first structure. But though it is stable, it cannot give an active protein. It cannot give a protein which functions much more efficiently. That means in the previous case, what we are producing, in the first case, we are producing a protein which is not that much stable in the environment. It is vulnerable to damage in the environment, but it can give rise to a very highly accurate, very highly active and efficient enzyme. But in second case, though it is stable, but it cannot produce any active or effective enzyme. That in this situation, in this kind of situation, cell always choose this protein which finally give a very active sequence. So in this case, cells are going to choose this first type of protein 3-dimensional structure rather than the second type of structure, because though it is stable, this is not at all about stability. If it gives a milder stability, that's fine for the cell will prevent that. But cell need much more efficiency, cell need much more activity, so it need much more active. So it might happen the several positions in the active site can be there, which can be mutated, that can give rise to more stable structure, but the structure will be less effective, like this second type. Okay, so active site of the enzyme and the binding site of the protein are really important, important for ours, and these are also the source of instability. So if we change any amino acid sequence, any one or two amino acid sequence in this active site of active site of the proteins, active site of enzymes, that will eventually going to change the whole function of the enzyme, the effectivity of the enzyme, okay, and the efficiency or work efficiency of the enzyme. Now we can also know that this protein structure can be destabilized using cold or using cooling temperature. So as we know, the denaturation can be done using high temperature. It can also be done using lower temperature. Now what this lower temperature is actually doing, lower temperature is actually hold those structure to close together. So minimum amount of oscillation is really important, because this oscillation helps this protein amino acid sequences to find their partners together to make bonds with each other. In in very low temperatures, what happens, this oscillation will be blocked. That's why in these situations those amino acid sequence will not be able to find their partners, and as a result they cannot make their bonds with each other. So that is how the cold higher cold lower temperature or cold can denature a protein. Okay, now what are the factors which are affecting the protein stability? As I've talked before, one is the pH, which is the proteins are the most stable at their isoelectric point, which they are not possessing either positive or negative pH. If they possess the positive site, if they possess the positive amount of acting, then there are different positive ions in the solvent which can disrupt their structure. If they possess negative, then there are negative ions which can disrupt the structure. But if they possess the neutral, there are no charges which can repel the backbone structure of the protein. Now the second thing is the ligand binding. Now most of the time ligand binding stabilizes the protein structure, most importantly in case of the enzymes. The enzymes in normal situations are stabilized, but when they bound with their substrate they become much more stabilized, and this stabilized structure will help the protein to maintain in this inside the cell. Now the third thing is the disulfide bonds. Now disulfide bonds are believed to increase the stability of the native state by decreasing the conformational entropy. Why? Because as we know, we are having the entropy increasing in the primary structure, then you can make a messy structure like that, which is called the molten globule. We are going to see in the later slide. Now then finally we are having the hydrogen bonds between these amino acid sequences, and after having those hydrogen bonds the structure become much more organized. So making this structure much more organized, this disulfide bonds actually stabilizing the 3D structure of the protein. Now the fourth things are the residues and the type of residues, and the most important thing about all those residues which you can find in 3D structure of a protein, the interior residues are playing the most important role, because the interior residues just place inside the cell. The interior residues are interacting with each other to make the core of the protein, and other residues are just surrounding the interior residues, and they are just facing themselves to the solvent, to the solvent which is water in this case. So these interior residues make much more what you can say contribution in making the protein structure stable, okay, rather than the exterior residues. Now if we think about the M value, there is another very important assumption in this case, okay. Now the thing is, the different M value, what is the M value actually? M value is the changes which can be used to understand the nature of the denatured state, okay. So that means the different M value related to the difference between the number of molecules of solvent bound in its native versus denatured state. Since of the folded state we have similar structure, the number of solvent molecules bound to the folded state is about the same. Okay, that reveals something extraordinary. Let me simplify this for you. The M value is actually telling us the number of amino acid residues which are free, which are denatured, or which are free, okay, in a simplified form. Now if we think the structure like this is a primary structure of a protein, which is amino acid interactions via the hydrogen bond, via peptide bonding. Then we suddenly then after all this we are having the 3D structure of the protein, which is a properly folded protein structure. Now if we think about this protein structure at the water, so if we put this protein, if we put this fully formed protein into the solvent in the water, then this protein is going to interact with water molecules, and we can also put this primary structure of the protein into water. They can also they are also going to interact with the water molecules in the environment. But by looking at this graph we can actually say that that by our actual practical knowledge we can say that when the protein structure is long in their primary structural arrangement, they can interact with much more water solvent than when they fold in 3-dimensional structure, right? But the answer is no. We can find the interaction of the solvent molecules are same in case of the primary, which is the unfolded state, and the folded state of the protein. Now the folded state of the protein will interact, suppose, with 200 water molecules in their environment, and if we giving the same environment and unfold the protein, this unfolded state of the protein is again going to interact with the 200 molecule of water, okay. So that is actually the basic concept. So that doesn't mean about the folding and unfolding. So if we change the folded protein to unfolded state, we can find the same amount of interaction with the solvent molecules that never varies, that never changes depending upon the changing of the state of the protein, okay. So that is the important part. Now we are going to look at the protein folding and some very basic things about the protein folding with the help of the Anfinsen's experiment. Now, Anfinsen is the pioneer who start to think about the protein folding in different ways, and he just conduct this experiment. He is having a native protein. He just took the ribonucleus as its starting point of the protein folding. So we have a native ribonucleus, and what he did in this case, he just added the 8 molar urea along with beta markup to ethanol. These are the disrupting agents. Now, beta markup to ethanol is going to disrupt the disulfide linkages among the protein subunits, protein domains and motifs. They are going to disrupt the disulfide linkages, and urea is going to disrupt the structure of the protein, other hydrogen bonds of the protein. Okay. It is going to stabilize the protein in its native form, like that. Okay, so they are adding all this. After addition of all this, what happens, the native protein is just getting denatured into something like that. Okay, it make this random coil. Okay, and this random coil shows no activity because this is not a folded protein. And as we know, protein folding is the most important thing about the protein function, because unless the protein is folded, protein cannot function. Okay, so after doing this, the protein structure will no longer active. Now, then what he did in this case, they denatured the reduced ribonuclease, and after having this denatured ribonuclease, they he dialyzes and remove all the urea, and also removes the beta markup to ethanol. And after removing the urea and beta markup to ethanol through the dialysis step, he just air oxidize all those sulfide linkages, and after that, the hypothesis was that all those sulfur will start to make the disulfide linkages with the help of air oxidation. And the hypothesis was proven right, and they get the native ribonuclease, and this ribonuclease is fully functional ribonuclease like the previous one. So what he did in this case, just took a ribonuclease, they disrupt the structure, they make it denatured, then again remove the causative agents which actually denatured this ribonuclease, and that eventually turns the ribonuclease into an active conformation. Okay, so after the denatures, it can also possess the same type of the same activity. So by looking at this, what we can conclude, that all the information that is necessary for a protein folding is not at all incorporated in the secondary structure, in the tertiary structure, or in the quaternary structure. Now all the information necessary for the protein folding is rather incorporated in its native structure, in its primary sequence. Okay, so primary sequence have it all. It's only dependence on the interaction. It's only on the interaction. When the interactions start to have, when the amino acids start to interact with each other, we are having properly folded protein. Okay. Now we are going to talk about the second step of the Anfinsen experiment, which reveals another conclusion, that after doing this, after using the 8 molar urea and beta markup ethanol to denature this ribonuclease, he go through another another round set of experiment. In this experiment, in this case, he is just removing only the beta markup to ethanol, but not removing any urea. Okay, just removing the beta markup to ethanol. And as we know, if we remove the beta markup to ethanol, that means those sulfur groups, those SH groups inside those threonine and serine residues in the amino acid sequence, they can interact with each other to make disulfide bonds. So they just remove this beta markup to ethanol, and what they can see the protein will make a structure of scrambled ribonuclease. This is not the ideal structure of ribonuclease that we used to find. This is a scrambled structure of ribonuclease. We can tell this is this is not the properly folded structure, but this is and we look for the activity of this scrambled ribonuclease, and this shows no activity at all. Why this is actually happening? We are removing the beta markup ethanol. That means we are enabling, we are make this this SH containing amino acid, we are enabling them to make bonds with each other, to make disulfide linkages with each other. They can make this linkage, but we are not removing the urea. That's why the protein structure will be freezed in this kind of situation. Okay, so that is a point. Now if we further addition of the trace amount of beta markup to ethanol is done, then what we can happen, then we can see the scrambled ribonuclease suddenly get a meaning. The scrambled ribonuclease finally make a structure which in turn is very much active like the original native ribonuclease. Okay, so so another important part is in this case is the native form of the protein has the thermodynamically most stable structure. That's why any nearer structure about this native configuration. So if we produce a near about structure of the native conformation, very near about structure, very similar structure of the native ribonuclease native protein conformation, but still the protein wants to go to its native structure. It's always a journey. It's always a journey for the protein. This is the take home message. It's always the importance of the protein. It's always print in the mind of the protein that we have to go to the native state. So whether we are going very close to the native state, it is 99% similar to the native state, but still they need to make this another 1% to make the 100% native state. So it is a journey from their native, from their scrambled state, from their unfolded states toward the native state where they belong. And this native state, and that's because the native state is the most thermodynamically favorable structure. Okay, so that's why this thing is happening all the time. Okay, and we can also think that these disulfide linkages are important, because when we just first remove this beta markup ethanol, the disulfide linkage is formed, but the formation of disulfide linkage is not the sequence, not the amount or the sequence we needed for a properly active enzyme. Now you can see in this picture, this enzyme after the removal of the beta markup ethanol can have this disulfide bonds, but the disulfide bonds form between 26 and 40 and 84 and 95, and those gives rise to the less effective and no active and without any activity of this activity of this enzyme. But if we add beta markup ethanol, then we remove all these things, then we remove urea, and then what we can have, then we are having again disruption of these disulfide linkages, and right disulfide linkages actually formed. So it is not about the linkages, this is not about only the interactions, hydrogen bonding, hydrophobic interactions or disulfide bridges formation. It's about the right things, formation of the right thing, formation of the right interaction. So for this ribonuclease, the bonding, the disulfide linkage between 26 and 84 amino acid residue is important rather than the 26 and 40. So if disulfide bridge is formed between the 26 and 40 residue instead of 26 and 84th residue, then it will make a structure which is no longer active. So it's not about only the making bonds, it about the specific bonding. So the bonding is specified to make the native state. So that's why the native state is really 1 in a million. We can find only one native state, because there are lots of amino acid sequences and there are lots of combination of amino acid can be done, but only one type of combination is favored, and that is the native combination. Okay, so that's why it's a very difficult journey to go from unfolded state to the native state, but proteins can achieve that. And for achieving this thing, there are some other proteins which actually help proteins to achieve that, because sometimes protein find this really tough to achieve. So in those situations they have proteins like chaperonin, which is actually helping the protein to achieve this very dangerous task. Okay, now let us talk about another important thing, which is called the Levinthal paradox. What just I've just talked about is, consider there just for the peptide backbone there are 3 conformations per amino acid. Okay, we know that there are 3 conformations per amino acid in the unfolded state. So suppose we are having only 100 amino acid sequence of a protein. So we have a protein which is a very small protein which is having only 100 amino acid sequences. We take this protein and we are having the 3 conformation of this amino acids. So we are having how many conformations? We have 3 to the power 100 conformations. Okay, now if the chain can sample 10 to the power 12 conformations per second, it take 2 into 10 to the power 28 years to reach and produce this native state of the protein, because that is really difficult, that is really dangerous. It cannot be happen. So it will take much more year, 2 into 10 to the power 8 years to achieve, to make a protein stabilized, to make a protein back to this native state or native conformations, because there are lots of conformations, there are lots of varieties of conformations that you can see. This is called the Levinthal paradox. And how this paradox is solved? The conclusion is that the protein folding is not random. that must have pathway. That is another take home message, just write it down. The protein folding is not random, because it cannot be random. If it has to be random, then it will be enormously complicated and very time consuming. So it have to have any pathway, and the pathway is always there. There is pathway, there is other proteins, there are other helper proteins which is helping the protein to achieve this very complex desire, and they can actually obtain that. Okay, so now we are going to talk about the molten globule state, and this molten globule state, which is the most important transient state for the protein folding. It is really important. Why? Because if we go from unfolded protein towards the folded protein, we are having the transition phase, and this transition phase is called this molten globule phase. Now this molten globule phase is nothing but the non-specific assembly of secondary structure. So we are having a protein structure where we are having the secondary structure fully formed, and the interaction of the secondary structure is less. The interaction of the secondary structure is lower, so there is no specific interactions between the secondary structure, though the secondary structure is being formed inside this protein, but the interaction, the specificity of the assembly of the secondary structure is none in this case. So we call this kind of structure a molten globule structure. That means we provide all those necessary domains, but we are not providing any specificity to the protein. As we know, the specificity is much more important, because unless the specificity, we can make a folded protein and sometimes they can be stable, but they can be they cannot have any activity at all. They cannot show any activity at all. Okay, so now let us move on and look for the kinetics of the protein folding. If we go from here, like unfolded state towards the folded state or native state, like that, and the transition state is the transition 1, this is the transition 2 state. And as we can see, if we look out about the kinetics of the folding, then we can say some very important consideration in this case is that not all the steps have the same rate constant. So the rate constant is varying from one step to another step, and the intermediates accumulate to the here in between this 1 and 2 intermediates is relatively small and relatively low in concentrations. So there are higher concentration of unfolded protein we can also find higher concentration of native protein in the environment, but we are having very lower amount of this transition state of the protein in the environment. That's another important thing in the mixture. So whenever look at the mixture, we are having unfolded as well as the native protein, but not the protein in the transient state, not the protein in the molten globule state. Okay, now if we look at there another very important assumption about that. If we are going from the unfolded state to the folded state, we are having much more unfolded state. We are having the unfolded protein here, and we have the folded here. Now if we look for an environment and look for the proteins, we can find the much more folded proteins rather than the unfolded state. So it's the lowest, this is the highest. So if we go from the concentration of protein, look at the concentration of different species about this protein folding, because if we journey from unfold, then we have the transition 1, then we have transition 2, suppose 2 transition, and then finally get the folded. Then we are having the highest concentration of the folded and the lowest concentration of unfolded, but we are having this transition state in between, but still these are the lower than the folded and unfolded state. Okay, so the unfolded state is an ensemble of large number of molecules with different conformations. So what we are talking about unfolded state, we have many different molecules, of the protein structure, many protein chains, but all those chains with different types of conformations, which is not the stabilized one, which is not the most stabilized one. That's why we call them the unfolded state. So we are having all those amino acid sequences with the peptide bonds and chains and all those things, but what we are lacking, the stabilization of the conformation, the specificity of this interaction between the amino acid sequences. That is the case about that. Okay, so here we are, if we look at the free energy thing, then we have the less free energy and the least free energy for the folded state. That's why it's much more favored. We are having the moderately high free energy for unfolded state, but we are having the highest free energy for the transition state. So we need to go through this kind of steps, okay, to finally make this folded state of the protein. If we reach at the peak of this transient state, then you can make this folding of the protein. Okay, So most of the time we are having the unfolding and the folded state, and less amount of this transient state phase of the protein can be found in the environment. Now if we look at the kinetics by this way, we have the unfolded protein. Now the unfolded protein, the amino acid sequences present in the unfolded protein start to interact with each other to make what is called a molten globular structure. This molten globular structure is nothing but the non-specific interaction between the domain of the proteins, and finally they interact with each other and finally make which is called a folded protein. In this folded protein, what we are having specific interactions of amino acid sequences in the domain to make a really compact structure like that. Now here we are having three types of model of protein folding. First model is called the framework model, second model is the nucleation model, and the third model is the hydrophobic collapse. Now we are going to see three different types of models, and now finally look what is the actual model to describe the protein folding. Now first model, or the framework model, what this framework model start to say is that we are having this primary sequence, stretches of amino acid linked together via the peptide bond. Now the region of this peptide amino acid sequence, or the primary amino acid sequence, start to make different domains. Some of the regions start to make the alpha helix domain, some of them start to make the beta sheet domain. Now making different domains, then what they do, they interact, those domains start to interact with each other. This alpha helix, beta sheets and alpha helix, all these things are starting to interact with each other to make the desired protein structure, to make the most stabilized protein structure. So what is happening, they actually diffuse until they coil. So they make this regional secondary structure, and this regional secondary structure start to diffuse and finally make the coil structure. Okay. Now if we look for the classic nucleation model, now this model states about the propagation of this secondary structure. So in this case, rather they make all the secondary structure that can be formed in the primary sequence, they can form only some stretch of secondary structure in some region of the this primary sequence of the protein. So it makes some secondary structure here. Now this secondary structure serves as a nucleus for this overall primary sequence of the protein. Now it acts as nucleus, and all the other strands, all the other sequence of amino acids start to accommodate onto this nucleus to finally make the coiled or actual three-dimensional structure of the protein. Okay, so this is called the propagation, because we start to make a nucleus, which is the starting point of the protein. Then we are arranging other amino acid sequences to the nucleus to make the strong and stabilized structure of the protein. And the third model is called the hydrophobic collapse model. And according to this hydrophobic collapse model, what is said is that there are regions in these amino acid stretches of sequences. There are hydrophobic amino acid there. Those hydrophobic amino acids start to interact with each other, because these hydrophobic interactions are really important inside the protein. So this hydrophobic interaction, so let me tell you what is a hydrophobic interaction. Hydrophobic interaction is a type of bonding that is done to go against the water, watery environment, or the hydrophilic environment. So the protein sequence, so that some of the amino acid sequence try to go away from water, and this driving force of go away from water helps them to accommodate with each other, to find their partners which are also hydrophobic, to interact with each other to make something, some stabilized structure which makes the interior of the protein most of the time. That is called the hydrophobic collapse. So according to the hydrophobic collapse model, so they start to make the secondary structure and would be directed, and so start to make this amino acid sequence to make the hydrophobic collapse, and after the hydrophobic collapse they will make the secondary structure. Then those secondary structure will interact with each other to make the tertiary structure. Now the tertiary structure will interact to make the quaternary structure. So these are the three different types of models that will explain how a protein is folding. Now I am going to tell you the classic mantra for protein folding, and that is that it is unlikely that there is a single mechanism for protein folding. So there is no single mechanism that can explain the protein folding. No single mechanism is there. So we can incorporate, we have to incorporate all these different types of mechanisms to describe how a protein is folded properly, because this is also, this is again is undescribed process. The research is going on on the full force to understand about this protein folding, because still it is unknown to us. The full mechanism is unknown to us, how it is initiated. But it is unlikely that there is only one mechanism of this hugely and highly complex mechanism. So there must be different ways to explain this. So we can accommodate all the ways, accommodate the nucleation model, the propagation model, which is nucleation, which is a propagation model, and also the hydrophobic collapse model, to finally describe this structure, describe the arrangement of the protein like that. Okay, now